on to today's lecture. My name is Uzma. I'm currently studying a Bachelor of Commerce, majoring in Accounting and Taxation at the University of New South Wales, and I will be taking the preliminary biology lecture today. I hope that you are all ready to learn some new information, hopefully, or get a little bit of revision as we prep for sort of the exams coming up for prelims. So this is basically going to be like a preliminary exam survival guide for you, hopefully. So yeah, I'm very excited to be able to take the lecture and hopefully go through that information from the 4 modules. So we'll be going through some revision and we'll also be going through an in-depth look at some modules. So yeah, well, before we get into that, I did want to go through some of the other resources, free resources and paid resources that ATAR Notes provide. So I know you've found this lecture, which is a free resource that ATAR Notes provide, but if you're not already aware, we've got a couple of other free resources that ATAR Notes provide. So we've got hundreds of downloadable study notes available, which is really great. So if you're struggling with a certain topic, then you can just sort of type in whatever the subject is and the topic, and then you'll be able to access that. We've got lectures where you can learn from the best. So this is the prelim biology one, but we've also got lectures for a range of other subjects. So if you are interested in having a look at some of those, feel free to do so. We've got discussions, so online Q&As where you can connect and discuss with your classmates, your peers, your cohort, your like tutors, things like that. Like tutors are on that as well. So if you're interested in like sort of having a discussion, you've got questions to ask, whether that be about university or HSC or something specific in your subject, that could be a great place to get that answered. Videos with engaging online revision, it will be really great to sort of break away from your notes. I know that when I had my notes or I just had the textbook and I was just reading all that information, sometimes it would be an overload and I would need something different to break away from just the traditional notes that you have on paper. So having that online engaging revision was so useful, like having those videos to break away and allow your brain to absorb information in a different way, and to also give your brain a break from just that pen and paper was really, really useful for me, and it might be for you too. So if that's you, that's something you can have a look into. We've got newsletters to help you stay in the loop, to know all the recent information. ATAR calculator, so if you've got a certain goal in mind for an ATAR, this could be really great for you because you will be able to see if you're on track, see what you need approximately for each subject, so then you can work towards that and hopefully get your dream ATAR to get into your dream university and degree. Articles, so study tips and strategies for like different subjects, things like that. So we have a whole range of different articles that you can have a look into and heaps more. So if you're interested in any of those, please feel free to check out our website, which is atarnotes.com/register for all of that information and more. So yeah, I would highly recommend to make use of some of those resources if you're interested. Okay, so now moving on to a couple of our paid resources. So these are all created by elite students to help you get the best marks that you possibly can. Right, so first of all, we have TuteSmart, which is an online tutoring platform from elite recent Year 12 graduates to help you do the best that you possibly can in your subject. So this online tutoring is great because we have options for private tutoring, so one-on-one, where you have your individual questions, you come in, you ask, we go through content, whatever it is that you want, we do for an hour, whereas we've got the class tutoring as well, which is in a group setting, and like for example, I do private tutoring for a range of subjects and then class tutoring for HSC business studies, so we've also got like a wide range of subjects. So if you're interested in that, or you need a little bit of an extra push for certain subjects, or you're not sure about certain subjects, you need a little bit of extra help, TuteSmart is the place to go. We've also got study guides, which you may already have seen. We've got them for a bunch of different subjects. It's printed revision materials with top tips to help you get the best marks that you possibly can. So I remember when I was in school, sometimes I'd look at the study guides and I would look at the textbook and go, wow, that is so thick. That's a lot of information that I have to retain, and it would be really overwhelming compared to the study guides, which were quite thin. There were a summary of the information, and it's super useful to help you get that revision in. And finally, Ed Unlimited, which is essentially the study guides that I was talking about all in one place online. It's like a Netflix for all of your study guides, so it's really, really great. So if that's you, if you're interested in that, if that's something that you think that, oh, that would actually be really helpful, have a look at these because they're really great. Like for example, the study guides, we've even got them for English for text specific subjects. If you're doing a certain text in English, we might have a study guide for it, and it's like with all the analysis and everything, so it's really, really great. So yeah, all of these resources are created by elite students to help you get the best marks that you possibly can, so would highly recommend. If you are interested in those, again, the link is down here for you to access that and register for it. Okay, without further ado, let's get right into the preliminary biology lecture for today, which is essentially going to be a preliminary exam survival guide. So hopefully this will be of benefit to you and that you can actually find some of the content that we go through helpful. So let's go through this. We're going to go through a revision for Module 1, which is cells as a basis of life, so cellular structures, movement of materials, biochemical processes, and enzymes. So we're going to go through a bit of revision for that, as well as for Module 2 we'll also be revising, which is the organisation of living things, so looking at nutrient and gas requirements, transport, etc. So we'll go through revision for that, so it won't be as in-depth, and if you would like in-depth, stay tuned because I'll provide a link to access in-depth sort of content for that one as well, for those two modules. And then for Module 3 is where our specific focus will be for today, which is biological diversity. So we're looking at things such as selection pressures, adaptations, theory of evolution, and evidence for evolution for that theory. So that's going to be our main focus for today. I always found Module 3 to be quite interesting to look at those different theories of what's been going on in our past and looking at biological diversity as well. And Module 4, which we will not be covering today because, yeah, we want to try and keep this between 2 hours. So, yeah, we've got 2 modules and then Module 3 as well. So go to atarnotes.com/notes/ for an in-depth look at the first 2 modules because we won't be covering it in as much detail. It'll just be a revision of some of the important concepts that students often find difficult, and with our focus today being Module 3. Module 1 overview. So Module 1 looks at cellular structures, arrangements, so looking at what's inside the cell, what's included in the cell. It also covers prokaryotic and eukaryotic cells. It covers technologies used to determine structure and function. Passive movement of materials in and out of cells, and active movement of materials in and out of cells, and how we can actually differentiate between the two is quite important, so we'll be covering that. Cell requirements, biochemical processes, and enzymes are all very important to cover and to go through. So we'll be going through all of that today, or not all of that, but most of that today as a sort of revision for Module 1. So, here's a snippet from the syllabus. So, investigate a variety of prokaryotic and eukaryotic cell structures, including but not limited to comparing and contrasting different cell organelles and arrangements. This is a snippet from your syllabus, and I'm going to actually teach you how to read your syllabus because I think that will be so important, especially leading up into Year 12, where you will have to learn this skill. So I'm going to quickly show you that now before we get into the actual content component. So number 1 is to read the working scientifically skills, right? So assess risks, consider ethical issues, select appropriate materials and technologies when designing and planning an investigation. These are the skills they want you to be able to gain from doing this component of the syllabus. Take notes, that's number 2. Take note of the inquiry questions. These are so important. Often students overlook them, but they are so important to success. So reproduction, for example. How does reproduction ensure the continuity of a species? That's the inquiry question. So that's the question that you want to answer. And number 3, pay attention to the wording. So evaluate using examples, the benefit of engaging in epidemiological study. So Here we can see, like they say, investigate. So you want to go into depth with that. Evaluate, what does that mean? If you're not sure what these verbs mean, would highly recommend going onto the NESA website, typing in HSC verbs glossary, looking at the definition of each of the HSC verb, so you know how to answer those questions. You know how to approach them when they come up. So, for example, investigate the response of a named Australian plant to a named pathogen through practical and or secondary sourced investigation. Right? So, for example, fungal pathogens, viral pathogens, etc. So they really want you to pay attention to the wording here. And these are tips, hopefully, that should help you to understand and to read the syllabus appropriately and then go from there. So hopefully that is making sense. So let's go through prokaryotic versus eukaryotic cells first. So eukaryotic cells are cells with a nuclear membrane and organelles. So for example, plants, animals, fungi, and protists all are eukaryotic cells. So we've got a diagram of a eukaryotic cell right down there. So we've got some of the features and some of the elements that are included in a eukaryotic cell. But essentially, we want to remember that it has a nuclear membrane and organelles. Right? When we compare that to the prokaryotic cell, it is cells with no membrane bound organelles. So prokaryotic cells are much simpler than the eukaryotic cells, and they are mostly unicellular, for example, bacteria. So you can see the comparison. It looks a lot more simple. There's a lot less organelles even than the eukaryotic, from what you can see in the diagram. So you want to be able to identify that and then be able to differentiate between them. So one has a nuclear membrane, one has no membrane-bound organelles. Okay, so that's the difference. And you want to be able to remember examples for each one when you are differentiating. So eukaryotic, plants, animals, fungi, protists, prokaryotic, bacteria. Okay, characteristics of a eukaryotic cell. So they have membrane-bound nucleus and membrane-bound organelles, like we talked about in the previous slide, right? So this idea that most of these are going to have a membrane compared to a prokaryotic, which does not have any membrane-bound organelles. So with eukaryotic, unlike prokaryotic, which just have unicellular or are just unicellular, eukaryotic can actually either be unicellular or multicellular. So organelles perform their specific function and carry out biochemical reactions essential for cell and organism function. Right, so it's important to take that into consideration when we are talking about eukaryotic cells. So this idea that they have specific functions, they carry out those biochemical reactions, and they're essential. So therefore, DNA for the cell can be stored in the nucleus. Okay, so that is the idea of a eukaryotic cell. Now, compare that to prokaryotic cells. These do not have membrane-bound organelles like we first talked about here. So there's no membrane-bound nucleus, unlike a eukaryotic cell. There is no membrane-bound mitochondria either. DNA is stored in the cytoplasm, and non-chromosomal DNA is stored in the plasmids. Okay? So what are some examples, like we went through earlier, for prokaryotic, there's bacteria, there's also archaea. So here's a bacterial DNA, the plasmids. We talk about the fact that non-chromosomal DNA is stored in these plasmids. Okay, so we've got the diagram here of the prokaryotic cell. We've got the key terms here as well, in case you were a little bit confused as to some of the terms I'm referring to. We've got organelle, which is an organized or specialized structure within the living cell. We've got membrane, which is a thin sheet of tissue or layer of cells acting as a boundary, lining, or partition in an organism. And membrane-bound organelle, which is an organelle enclosed in a membrane. So those are the three. Organelle, organized or specialized structures within the cell. We know that we've seen some of those. Membrane, which is looking at a thin sheet, that membrane-bound organelles, those membrane-bound organelles which don't exist in the prokaryotic cell, whereas we do see them in the eukaryotic cell. And yeah, so that essentially is prokaryotic cells, and make sure you do know examples of each. So you know examples of a eukaryotic cell, you know examples of a prokaryotic cell. So, looking at the comparison between the two, between a prokaryotic and a eukaryotic cell. So, prokaryotic cells, for example, have the similarities in that they both have DNA, right? Prokaryotic and eukaryotic cells, they both have DNA, they both have a cell membrane, and they both have cytoplasm, which is the fluid within the cells. So both of them have those three features: DNA, cell membrane, and cytoplasm. What are the differences? Differences in the prokaryotic versus the eukaryotic. So prokaryotic is more simple, like we initially mentioned. There is no nucleus to contain DNA, so therefore DNA is free floating within the prokaryotic cell. And as we talked about earlier, there is no membrane-bound organelles. Okay? When we compare that to the eukaryotic cell is a lot more complex. There is nucleus to contain DNA, unlike the prokaryotic where it's free-floating. And finally, unlike a prokaryotic cell which has no membrane-bound organelles, a eukaryotic cell does have membrane-bound organelles. So those are the key differences between them, whereas the similarities are, again, DNA, they both have a cytoplasm, and they both have cell membranes. So that is how you can differentiate between them. If you were asked in an exam now the difference between a prokaryotic versus a eukaryotic cell, hopefully you would have enough information to be able to identify that difference and distinguish between them. So here's a bit of a summary. So synthesis and processing of proteins and lipids. So nucleus stores genetic material and coordinates the cell's activities. So that's growth, protein synthesis, and cell division. So the nucleus plays a really important role. We've got a ribosome, which acts as the site of protein synthesis, and it's found on the rough endoplasmic reticulum. So remember, there are two types of endoplasmic reticulums, or ERs. We've got the rough one and the smooth one. So the rough ER transports proteins produced by ribosomes, like we initially talked about, into the Golgi apparatus. Whereas the smooth ER is the site of synthesis for phospholipids. Okay? So I know that these might be a lot of new terms for you if you haven't already gone through these, but make sure that you do understand the role of each one. So we've got the Golgi apparatus, which looks at packing proteins into vesicles, modifying them, and then sending them to their destination, whatever that destination may be. Lysosomes digest essentially unwanted matter, such as damaged organelles or foreign material. They're there to sort of digest that and then identify and digest that, essentially. We've got storage and structure of cell structures. Storage and cell structures. So, vacuoles, they provide structural support in plants, specifically. So they're there, again, for that idea of storage and waste disposal. Plastids, which contain genetic material and play a role in the synthesis and storage, so not found in animals. Cell wall, this is the rigid structure that's found in plants, and that provides structure for the cell. So it's in the name, really, cell wall. Cytoskeleton supports the cell structure and movement and assists transportation within the cell. Centrioles are involved in cell division. And finally, the cilia and the flagella are both hair-like structures involved in the movement of cells. So, we can now see that we've identified some of those, so that you can see actually some of those. Smooth endoplasmic reticulum, the ribosome, the lysosome. You can see some of these now that we've been talking about. Rough endoplasmic reticulum, the nucleus, right? And we can see here the cytoplasm. Ribosomes, nucleoid, which is essentially DNA. Capsule, plasmid, etc. So we can see some of those, and hopefully that's now starting to click. Finally, the energy transformations. A lot of people already know of the mitochondrion. It is the powerhouse of the cell. It plays an important role in the respiration and the conversion of energy from food into energy used by cells. And finally, chloroplasts, which play an important role in the photosynthesis in plants, trapping light energy. You may have already heard of chloroplasts and photosynthesis because you may have gone through it earlier. But yeah, this is essentially sort of broken down in accordance to what their role is, and hopefully that should make it a little bit easier to sort of identify what each one does and what each one means. So yeah, that's essentially the cell organelles and their different functions. So hopefully that's all making sense. Okay, moving on now to organelles and their cellular arrangements. So we have some things that we need to identify here. So this is again a little bit of a summary of what we went through in the last slide, but in a new format. So we'll go through some like more examples of it to hopefully allow you to get an idea of what I'm talking about here. So energy transformations, we know that we've got the mitochondrion, which is the powerhouse of the cell, plays an important role in the cell respiration and converting food and into energy used by cells like we talked about earlier. So again, this is an energy transformation. What is the transformation? We're getting the food, we're converting it into energy that the cells can then use, right? To complete whatever function they are completing. Now, chloroplasts, like we talked about on the last slide, plays an important role in photosynthesis because, which is in plants, and the way that they do this is they trap light energy and then use that. So those are energy transformations. Make sure you're aware of what's involved in energy transformations, what those transformations are, and then we can go from there. Okay, so that is the first part. Now let's go through cellular respiration as an example. So respiration is the process by which cells use oxygen to break down sugar glucose to obtain energy. So that is going to be our definition of how cellular respiration will occur. Okay, so again, cells use oxygen to break down sugar glucose to obtain energy. Now, there are two ways that this can be done. Aerobic. So aerobic respiration is carried out in the presence of oxygen. So 36 ATP are produced as a result, versus anaerobic. So the way that you can remember it is an means no oxygen, because respiration is carried out in the absence of oxygen, where 2 ATP are produced. So aerobic is with oxygen, anaerobic is without oxygen, and aerobic produces 36 ATP, and aerobic produces only 2 ATP. So making sure you're understanding those differences, making sure you're understanding how respiration occurs. Again, looking at the use of oxygen to break down sugar. Okay. Then we have photosynthesis, what we were talking about with chloroplasts, even in the previous slide. So photosynthesis is the process by which plants use light energy, like we talked about. They trap the light energy to produce their own food in the form of glucose. So light energy transforms to glucose, which is their form of energy. So this is carried out in two specific stages, like we had with aerobic and anaerobic in cellular respiration. In photosynthesis, this is carried out in two stages, which is the light dependent and the light independent stage. So the light dependent stage is photolysis, which occurs in the grana. So it's really important to be able to identify where it occurs as well, and the two types. So make sure you are able to correctly identify that. So light dependent stage occurs in the grana. Light independent stage, which is also known as carbon fixation, it occurs in the stroma. Okay, so you want to be able to identify where they sit in the actual cell. So again, a little bit of a summary of this slide, because I really want you all to be able to really understand in detail what I'm talking about here. Mitochondrion and the chloroplasts are the two energy transformations. What is the one for mitochondrion? They're converting energy from food into energy used by cells, and that's how respiration occurs. Cellular respiration, We have cellular respiration, which is the use of oxygen to break down sugar, also known as glucose, to obtain energy. How is this done? It's done by aerobic and anaerobic. So aerobic produces 36 ATP, anaerobic produces 2 ATP. Photosynthesis, we've got the process by which plants use light energy to produce their own food. What's their food? Glucose. So that's the phloem. Again, it's carried out in 2 stages, the light dependent and the light independent stage. What's a light dependent stage? Also called photolysis, independent called carbon fixation. Where does a light dependent stage occur? In the grana. Where does the light independent stage occur? In the stroma. Okay, so that's a quick summary again of the organelles and their cellular arrangements, and we've gone through examples now of cellular respiration and photosynthesis. So, next we're going to investigate the way in which materials can move into and out of cells, including but not limited to conducting a practical investigation, modelling diffusion and osmosis. Again, think back to how to read the syllabus. You need to be able to identify what's involved, be able to identify the terms. If I go back over here, so read the working scientifically skills. Then you want to take note of the inquiry question and pay attention to the wording. These 3 steps are so important to success in this course, right? You want to be able to identify how to accurately read the syllabus, how to correctly read it, and how to correctly identify and approach it when you get to the stage where you want to be able to write exam answers that conform with the syllabus. So, passive movement of materials. So there are 3 types, right? Diffusion, osmosis, and the concentration gradient. Make sure that you know these and you know how they work. So diffusion is the passive movement of materials from high to low concentration until an equilibrium is reached. Osmosis is the passive movement. These are all going to be passive movements. Give me a sec. So diffusion again, high to low concentration until equilibrium is reached. Particles. Osmosis is the passive movement again of water from high to low concentration across a semi-permeable membrane until equilibrium is reached. And finally, concentration gradient, which I have got the diagram over here, is the passive movement of water from a high to low concentration across a semi-permeable membrane. So you can see the differences between them, right? So you want to be able to identify the differences between diffusion, osmosis, and the concentration gradient. So we've got the example of concentration gradient here. So remember that it's the movement of water from high, so over here, all the way to low concentration across a semi-permeable membrane. Osmosis, we have 3 types of solutions: the hypotonic solution, the isotonic solution, and the hypertonic solution, right? So here's the diagram as well. So we've got the different areas conducted for osmosis. So again, be able to identify diffusion. What is it? It's a passive movement of particles from high to low concentration until equilibrium is reached. Osmosis, passive movement of water from high to low concentration across a semi-permeable membrane. Again, until equilibrium is reached. And concentration gradient, passive movement of water from high to low concentration across a semi-permeable membrane. Those are the 3 for passive movements that you want to be able to remember. Moving on to active movement of materials, so active transport. The process requires ATP, which is energy, like we talked about earlier in respiration. So needed to move against the concentration gradient, which we have over here. So we need to now move against it, which is going to be a little bit different to passive. So maybe needed if a substance is actually charged or it's too big to be able to originally diffuse, which is a process of diffusion, right? So they can't actually undergo that process. So then we have active transport that occurs, and there are 2 types: endocytosis and exocytosis, and the way that you can remember them is through the names, which I'll talk about in a second. So before we get into that, how does endocytosis occur? It's when particles are too large, membrane changes its shape so that it can surround and engulf a particle and take it into a cell. Now this is known or an example of this rather, is phagocytosis. So again, it will change its shape, surround, and engulf. Okay? Exocytosis, however, is a little bit different. It is where membrane-bound vesicles move to the cell membrane, fuse with it, and then release the contents into the environment that's surrounding it. So transport substances out of the cell that are needed elsewhere or are in too high of a quantity in the cell. Okay? So again, one of them is moving in, one of them is releasing, or it's going out. So endo in, exo out. Okay? That's how you want to remember it. That's how I remembered it. So you can see the diagram over here. See the extracellular fluid. You can see the inside of the cell. That's where we see active transport occur. And there's the diagram example there as well. Right, next up, we've got conducting a practical investigation to model the action of enzymes in cells. So, enzymes are highly specific biological catalysts that bind with specific substrates called the activation site. Right? So the way that we will look at this is through the lock and key model. Right? So we can see those steps. I won't go through each step. Actually, I might just go through it. It's actually fun. So we've got the bond. Substrate is sucrose, which is composed of glucose and fructose that's bonded together. So the substrate will then bind to the enzyme. And you can see it's a lock and key model. And that's where the name comes from, as you can see, where it fits into the lock and key. Number 3, bond binding, the substrate breaks. Number 4, the products are released. So you've got the fructose, you've got the glucose. And number 5, the enzyme is free to then bind with other substrates. Okay? So you can see that enzymes movement, and then you can also see the movement of the substrate. Yeah. This is a little bit different from the induced fit model, where first you have the substrate again of sucrose, which is composed of glucose and fructose, that stays the same. So substrate binds with the enzyme, but it's an induced fit. So the binding of the substrate induces the enzyme to change the shape to fit more tightly, rather than the lock and key. And then the steps are the same after that. Okay, so that's the difference. Make sure you understand the difference between a lock and key and induced fit. You can see the difference. Like induced is like it's sort of changing around to fit, whereas a lock and key is just sort of happening immediately. That sort of concludes our overview of Module 1. So it wasn't in too much detail, but I just wanted to still have that in there for you all to access and to provide a bit of a summary of it. We'll now move on to Module 2. Where we're going to look at nutrient and gas requirements. So we're going to look at the xylem and phloem. We're going to look at respiratory systems in different organisms and circulatory systems in different organisms as well. Okay, moving on to the gills. So the paired respiratory organ of fish and some amphibians by which oxygen is extracted from water flowing over surfaces within or attached to the pharynx. Right? So we're looking at the walls of the pharynx and we're going from there. So water flows into the gills, into the gill slits in the side of the fish. So if you've seen a fish before in the structure of it, you know that they've got gills on the side. And then deoxygenated blood moves into the secondary lamellae in preparation for the water to flow in. And then we've got oxygen is then absorbed from the water into the lamellae, which moves into the blood, and then carbon dioxide is released. Okay, so like in humans, blood is then pumped back in to the fish using the heart. So in that way, it's similar to humans. We've then got the high assay increases the rate of the gas exchange. So that is a macroscopic gas exchange. And we can see the diagram there of the gills. Moving on. We've then got the trachea. So we're looking at a large membranous tube extending from the larynx to the bronchial tubes and conveying air to and from the lungs, the windpipe essentially. So that is another form of gas exchange when we see that macroscopy. We can see outside insect and inside index. We've got the trachea, we've got the spiracle, and we can go from there. So, gas exchange occurs directly onto the cells of an organism versus closed. Okay, so blood and interstitial fluid, so the fluid that surrounds the cells, is pumped into different vessels. So, in insects, air flows through a small opening in the body called spiracles. Right, so that's where the actual air comes in from. It then goes into the trachea and then into the small branching structures. Okay, and we can see them here. So it goes into the trachea here, and then it goes into the smaller structures that you can see here. Into the cells, the tracheoles branch into every cell in the body, allowing oxygen and water to diffuse in and carbon dioxide to diffuse out. Okay, so the movement of air occurs through passive diffusion, like we went through over here. Passive movement of materials, diffusion. And or active ventilation, where larger insects contract their internal organs and abdomen to force air out. So it just depends on the size of the insect as well. So something to consider. Something to consider there. Digestive system. So we've got the mouth, which is physical digestion. So teeth chew, tongue shapes food, increases assay for chemical digestion. So the enzymes sort of break down the starch. They sort of begin that breakdown. So enzymes are known to break down that starch. And then we've got the esophagus. We've got the bolus. So we've got the breakdown of starch then continues through. The epiglottis then blocks the respiratory system. We've got the stomach. We've got, of course, physical digestion. So sometimes you hear your stomach churning. That's what it's doing. It's digesting. It's relaxing. You've got gastric juices that combine with the food. And then you've got macromolecules, which are broken down into their building blocks. We've got the small intestine, which we move down into the small intestine. And what we've got is 3 regions. So the chyme from the stomach causes the release of pancreatic juices, and the large intestine aids with the absorption of the nutrients and converts the food, and then it comes out through your feces. So beneficial bacteria in the large intestine actually helps to digest these substances, and some substances such as water and ions are released and used for metabolism. So that is sort of a summary of what is actually involved in the digestive system. So hopefully that is making sense. Okay, so the remainder of the digested material will then, from the small intestine, which is over here, which is over here, will then move into the large intestine, which is over here. So this includes water, salts, dietary fiber, whatever. So vitamins A and K are then produced by the large, also get absorbed into the bloodstream. So there you go. That's the sort of summary of the digestive system. Moving on to the xylem structure. Xylem's vessels are long water-filled tubes consisting of elongated cells joined end to end. So as the cell matures, the cell wall is strengthened with lignin, which makes them more rigid than they were. So over time, when it matures, it sort of gets more rigid. The cytoplasm and nucleus in the xylem vessel cells then disintegrate and the cells die. Right, so creating hollow lignin tubes. Mature xylem vessels, so perforated or complete openings at each end, kind of like a straw, so that fluid can flow directly through it. So we've got pits, which are unthickened areas, and perforations in the side walls allow sideways movement of substances between neighboring vessels in the vascular bundle. Right, so no nucleus or cytoplasm exists here. So the tracheids are single large tapering water-filled cells that form part of the xylem tissue in all vascular plants. So when mature, they lose their nucleus and cytoplasm. So this leads to the cell death, but creates an open structure for water to flow through it. Then we've got mature tracheids, so cylindrical skeletons of dead cells actually join together to form continuous tubes. So unlike xylem vessels, tracheids are not connected end to end. And so instead their ends overlap and water is transferred horizontally through adjoining pits. So those are some of the key terms that we need to understand in the xylem structure. Moving on to the phloem structure. So phloem tissue is composed of the sieve tubes, the companion cells, the parenchyma cells, and the sclerenchyma cells as well. So we've got the phloem structure, as we can see over here. Unlike xylem cells, mature phloem sieve tubes are living cells. So they with no nucleus or no lignin in their cell walls. So that's a difference, right? You want to understand the difference between the two because you may be asked that in an exam. Sieve tubes form linear rows of elongated cells. Their cell walls are thin and perforated at each end by holes or pores, which form sieve plates. So the plasmodesmata pass through the perforations of the sieve tubes, and they act like straws through which water and other materials can actually move. So it's very interesting how it actually differs from the xylem structure and what the different elements are. So let's go through definitions. So sieve tubes, they provide structural support, work with companion cells to transport the molecules, as we talked about. Companion cells, they enable transportation of these sugars. Then we've got the parenchyma cells, which provide structure to the vascular tissue. The sclerenchyma cells provide structural support to the actual phloem, and the plasmodesmata cells enable the transport of the actual molecules. So each one has a structure, and it's important to memorize and understand what each structure is and how to approach it. So macroscopic structures in animals, looking at fish. So single circuit of blood flow. So blood flows from the gills straight to the capillaries rather than passing through the heart, versus in mammals who have a double cycle circulatory system. So understanding in different types of animals what the different types of systems are. So the heart has two chambers, so blood is pumped to the gills for gas exchange versus lungs. So this enables fish to regulate one circuit of blood flow. So you can see gill capillaries, the heart, the body capillaries, the water and the blood within the fish. It's a single circulatory system compared to the double. So open circulatory system, this essentially means that they have dissolved nutrients that travel directly to tissues rather than remaining in blood vessels. Right, so the heart, you've got back end of the dorsal vessel, okay, divided into the chambers called the ostia, and the contract to push it forward. Okay, so we can see the dorsal vessel as a tube running longitudinally in the insect, which is responsible for transporting the material from the abdomen to the head. And then we've got the aorta, which is the front part of the dorsal vessel, okay? Tube leading to the head where it empties, and then we've got after emptying, it freely travels through the organism. So it's important to have a look and sort of see. So we've got the ostia over here, the heart over here, the dorsal diaphragm over there. So just understanding where everything is in the insect as well. Alrighty, so now that we're officially done with Module 2, an overview, we'll now move on to Module 3. In Module 3, we'll be covering quite a bit. So we'll be going through effects of the environment on the organism, adaptations, the theory of evolution by natural selection, and evolution, the evidence. Okay, so 4.6 billion years in 1 hour. Yeah, there we go. Like, basically, this idea of all of the things that are involved at which times. So it's very interesting. Let's go through the peppered moth. So during the Industrial Revolution, the moth population changed from majority white to majority black. And a lot of people were like, whoa, how did this actually happen? And the theory is, well, what this is trying to say is that it was due to a change in their environment. So there was increased pollution and smog. So obviously during the Industrial Revolution, there was a lot of pollution, manufacturing, etc., and hence caused a lot of smog. So number one, within the moth population, there was always variations, as some were mutants born coloured black in a predominantly white population. All right, so you have some black and some white. So environmental selection pressure was applied to the population due to the change in environment because of the pollution. So birds could better spot white moths as prey and eat them than the black ones, which was then, camouflage. Most white moths were killed by predators, whilst the white moths actually survived because they were able to camouflage. As a result, moths with the black phenotype or physical structure reproduced and passed on those colorings to their offspring, and after many generations actually achieved the dominant trait. So that is sort of the diagram there as well. Okay. So biotic versus abiotic factors. So biotic factors relate to anything that's living, whether that be organisms living in the same ecosystem, competition for resources, so food, territory, predation, disease, etc. Whereas abiotic factors relate to anything that is non-living. So, for example, climate, temperature, wind, shelter, availability of food and energy sources, so access to water, light, essential nutrients, these are all non-living factors. Pollutants, chemicals, pesticides, insecticides, all of those things are non-living. So just understanding the difference between the two if you were asked that in an exam. So, cane toads, introduced to Australia. Cane toads were introduced from Hawaii to North Queensland in 1935 in an attempt to control populations of native beetles, grey-backed cane beetle, which ate sugarcane crops without using pesticides, because pesticides would also impact the crops, right? So spread. Cane toads started to spread rapidly, reaching the NSW border in 1978 and the Northern Territory border in 1984. The current population is estimated to be around 200 million approximately. This has resulted in the widespread ecological effects of the spread of disease, for example, the loss of biodiversity, spread facilitated by traveling on clear roads. So they would literally travel on clear roads all the way to these locations that were very far away. They were hitchhiking, building and landscaping materials, things like that. Evolution. So the population has rapidly changed over the last 70 years due to special sorting. So toads naturally born with longer legs move further and they breed with outer toads who have also traveled far. So the genes for faster movement, when their genes are passed on, become concentrated at the forefront of the invasion, and they're passed on to the offspring who become really fast as well. This has allowed rapid evolution over 50 years. They used to travel 10 kilometers a year, and now 55 a year. So it's very, very, very fast, right? Compared to what it was. We can see that evolution occurring. Population changed. So cane toads on the front line of the invasion have longer legs, are more agile, have a reduced immune response as less energy is invested into it, in preference for consistent long distant movement. So cane toads can also rapidly acclimatize to colder climates by behavioral adaptations. So, for example, by basking. And rapid psychological shifts. So this will they have a lot of adaptations to adapt to different weathers, different climates, things like that, so that they can actually spread even further than what's anticipated, and they can travel very, very far. Next, we're going to conduct practical investigations individually or in teams, or use secondary sources to examine the adaptations of organisms that increase their ability to survive in the environment, including structural adaptations, physiological, and behavioral. So, adaptations. What is adaptation? It is simply a process by which an organism, or species becomes better suited to its environment. So, structural adaptation, for example, adaptations that form part of the physical features of an organism. So, physically, there's been a change. So, for example, rootless teeth which regrow in wombats. Physiological adaptations. So, adaptations which result in a chemical change rather than a physical change to the body of an organism. So, sunflowers grow towards light sources. That's an example of a physiological adaptation. Next up, we have behavioral adaptation, so changes to organisms' behavior in order for them to live in their environments better. So, kangaroos, for example, lick their forearms in hot weather. That's an example of behavioral adaptation. Structural, physiological, behavioral. Next up, let's investigate through secondary sources the observations and collections of data that were obtained by Charles Darwin to support the theory of evolution by natural selection. For example, finches in the Galapagos Islands, as well as Australian flora and fauna. Know these two examples, they're in the syllabus, which means you need to know them. So we'll go through them. So Charles Darwin was an English naturalist, developed the theory of evolution by natural selection. So where's his voyage? You can see it sort of goes through Australia, went through a bunch of other places as well. So Charles Darwin joined other scientists on the HMS Beagle to travel around the world and collect natural history and geological samples to try and prove his theory of evolution. Okay, introduction to the theory of evolution by natural selection. So, change in a species over many generations as a result of natural selection of favorable characteristics is this theory. Evolution is a gradual process. It cannot happen overnight. It happens over many, many years, and it's facilitated by the mechanism of natural selection. So, steps of evolution. How would this have occurred? First of all, there's a variation that occurs within the population due to natural mutation. So think back to variation of the moth. So that one was black, or there were a few black ones and a few white ones. Selection pressures are applied. So physical change, chemical change, competition. So changes in biological niches are adapted or inhibited. Sorry, the second one should be where we say black or white moth. Phenotypes are best suited to the environment survive. So think back to, okay, the black moths were more likely to survive because they could better camouflage, and the white moths couldn't because the birds would eat them. Or in this sense, certain finches will develop suitable beaks and consequently survive as they can still eat. Surviving organisms then reproduce, and then they pass on those traits, which is a very important point. They pass on that trait to the majority of the population. What is a selection pressure? I've been saying this term, but if you're unsure of what it is, it's an external agent which affects an organism's ability to survive in a given environment. So, investigate through secondary sources the observation collection of data. So, we'll go through finches of Galapagos Islands and then explain using examples how Darwin and Wallace's theory of evolution by natural selection accounts for convergent and divergent evolution. So, we'll go through that now. So, different evolutionary processes. So, convergent evolution, this is the process by which organisms which are not closely related evolve to have similar traits because they are adapted to a similar environment. So, similar environments means that they have similar suitable traits, right? So, they sort of essentially grow to have similar traits because they're all in a similar environment. Whereas divergent evolution is a process by which organisms which have recent common ancestors, so they're closely related, evolve to have different traits, right? So that's a difference because they have adapted to different environments. So they have a common ancestor, explained by Darwin's theory through the example of the finches from the Galapagos Islands. So, why Darwin's finches carry the entire biology syllabus? Let's go through it. So, why Darwin's finches is better than every other case study ever? Because there's just so much information provided. So, the evidence. So, Darwin's finches are an example of gradual speciation. There are 14 species diverged from the common ancestor of the mainland finch. Okay, mainland finch. Darwin discovered 12 of these species, which had never been discovered before. Finches developed different beaks sizes and shapes to suit the diets in their individual biological niches. So we've got some fruit eaters, insect eaters, we've got some seed eaters. Their beaks were sort of shaped accordingly. Why the finches make the biology course? They actually demonstrate how species adapt as a result of selection pressures, which is something we went through earlier, the external agent, remember? External agents which affect an organism's ability to survive in a given environment. Remember that definition? That's why it's here. They're a great example which can be used for multiple points in the syllabus, which is why I would recommend making sure you do know this example. So, Darwin, Australian flora and fauna. So, instead, this was an example of convergent evolution, where unrelated animals adopt similar features due to similar selection pressures. For example, Darwin noted that the Australian platypus behaved in a similar fashion to the European water rat. Okay, next up, analyze how an accumulation of microevolutionary changes can drive evolutionary changes and speciation over time. For example, the evolution of the platypus. So, platypuses are believed to have diverged from the following lineages. So, we've got due to a blend of characteristics. So, we've got platypus, we've got reptiles which have venom, birds which are egg laying, and mammals which have lactation features. All of these are present in the platypus. Evolutionary change. So, this refers to the genetic change in species over time in response to different environmental selection pressures. So, this change is due to the process of natural selection, which involves Darwin's 4 points. So, these 4 points are proposed by Darwin. So, we've got microevolutionary change. So, evolutionary change within a species or small group of organisms, especially when it's over a short period of time. So, that's why it's called micro, because it's a small group or a short period of time. Macro is looking at a combination of microevolutionary changes which can cause speciation, so the formation of an actual entire new species. Macroevolutionary change. The platypus. Species are a classification of a group of closely related organisms which possess very similar characteristics, who can breed and create fertile offspring. That is a species. So let's go through macroevolutionary change. Another example is of the horse. So we're looking at the analysis of how an accumulation of microevolutionary changes can drive evolutionary changes and speciation over time. So, for example, looking at the evolution of the horse and the platypus, this one's the horse. So you can see 50 million years ago versus 35 versus 26 versus 3 million, and how it's actually changed. How the forefoot and the skull has changed over time, and the length of it as well, going from 38 centimeters to 52 centimeters to 100 centimeters to 135 centimeters. So, the first type, the dawn horse, it's likened to a dog rather than a horse. So, what we would consider a dog today, kind of like a dog shape or whatever, is what that was. 4 toes on each front foot and 3 toes on each back foot. And they have omnivorous teeth. About 30 centimeters tall. And you can see the forefoot there. Then we have the next type, which had more grinding teeth, and it was to suit a plant-based diet. Okay. Next up, we have the next type of horse, which had even tougher grinding teeth and larger with longer legs, aiding with speed in open areas. So we can see that they've got the forefoot there as well, and the difference between the third to the second and the first. Finally, we've got the last type, and they've got hypostodont teeth. So teeth which grew continuously out of the gums as they were worn out. So developed jaws with ridges for efficient grinding, and long legs and body size aided in running in open areas. So those are the 4 different, steps, and you can see those steps of how the horse has changed over time to create macroevolutionary change of the horse. So the equus, or the modern horse, looks at this idea of the horses beginning to evolve with singular toes, strengthening their toe ligaments, and removing the side toes, which may not be as much used. So they're not really using them as much. So they're trying to. So over time, this sort of disappear. Appeared around 4 million years ago and is what we identify as the modern horse that we see today. What are the features? Long legs, long necks, horse-like body, deep jaw, and long faces. So that is the macroevolutionary change of a horse, and then we saw the macroevolutionary change of the platypus as well. So this idea of lineages and the different characteristics being developed and put into that new fertile animal. And we saw it with the horse. Now we're going to move on to punctuated equilibrium. So, explain how punctuated equilibrium is different from the gradual process of natural selection. Let's do it. Until the mid-20th century, evolution was considered to only occur by the gradualism Darwin suggested, with slow changes in a species over time due to natural selection. Alright, so this idea that it was a slow change, right? However, in 1972, Eldridge and Gould instead suggested that evolution is far less consistent and gradual as Darwin theorized. Punctuated equilibrium is believed to be a period of rapid evolutionary change, leading to the emergence of a new species, followed by a period of stability and stasis where the population remains unchanged. So gradualism, we can see here. Whereas punctuated equilibrium, we've got the changes, and then for a long time, they're sort of stable, they stay as they are, and they're not changed. So just understanding the difference between gradualism versus punctuated equilibrium. So, punctuated equilibrium, we can see the evolution in time and we compare that to the theory of evolution. So, theory of evolution says new species formed, new species formed, new species formed over time, like it's as time goes, like it is just new species being formed. Whereas punctuated equilibrium says, wait a minute, that's not exactly how it works. It's this idea of we have evolution, we have time, we have a new species, but there is stability in that process and we're sort of stable for a while before we see a new species again, and then again we're stable for a while, and then we see a new species, we're stable for a while, and that process continues. Rather than just new species, new species, new species, punctuated equilibrium takes some more conservative approach and says, actually, no, let's calm down, let's have a period of stability again, then we have a new species a little bit later. Okay, so the theory of evolution is sort of go, 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 whereas punctuated equilibrium sort of goes calm down. Okay, let's sit down, let's take a break. Okay. Moving on to amino acid sequencing. So, where is this in the syllabus? Investigate using secondary sources evidence in support of the Darwin and Wallace theory of evolution by natural selection, including but not limited to biochemical evidence, comparative anatomy, comparative embryology, and biogeography. So, amino acid sequencing, two organisms sharing part of their amino acid sequence, is strong evidence for divergent evolution. Going back to divergent evolution, remember what we went through over here. Remember the process by which organisms that have recent common ancestors but they have adapted to different traits in different environments. The horse, yeah, it's fine. Here. So amino acids are the building blocks of proteins. So it's a really great way to figure out if these, you know, like to look at the amino acids and compare and see if the divergent evolution has actually occurred. So how does this actually work? Because it doesn't sound, it sounds really like quite a lot to actually go in and see it. And you can see in the photo, it doesn't look quite as simple as we thought. Scientists compare amino acid sequences which make up specific proteins of different organisms. For example, the cytochrome C, the hemoglobin, etc. So we can see here the phylogeny based on nucleotide differences in the gene for cytochrome C. So humans, monkey, donkey, horse, dog, horse, donkey, pig, rabbit, kangaroo are sort of having a similar wavelength. And then we've got the pigeon, duck, chicken, penguin, turtle, all in tuna, all in the same sort of wavelength. And then we've also got like a bunch of other ones there as well. Organisms which share part of their amino acid sequence. So we've got molecular homology of cytochrome C. So amino acid position 1, 2, 3, whatever. And we've got the human, chimpanzee, pig, chicken, dogfish, wheat, yeast, etc. And we can see that. Right, so the source is from the Year 11 Biology in Focus. Now moving on to biochemical evidence looking at DNA and DNA hybridization, which allows us to determine relatedness through comparing nucleotide base pair order of different species. What is this process? So we've got the double helices of the DNA from two different species, which are split when you've got the 1994 heat applied. The separated segments of DNA from the two species are then mixed. You can see here, got the species A, species B, then they are mixed together. These mixed strands then combine to form a hybrid DNA molecule in a process called reassociation. Closely matched base pairs will strongly bind to one another. Okay? So if they strongly pair to one another, heat is applied to separate the strands once again. The higher the temperature, the closer the bond. The more nucleotide base pairings which are closely bonded means that they are more related they are as a species. So we can see over here sequences similar and they are very difficult to separate, right? Because they're all connected, you can see, compared to this one which is not connecting. This means that species A and C are closely related. compared to species A and B, where you can see that they are actually not connecting and they're dissimilar and they're very easy to separate, meaning that they're not closely related. Moving on to comparative anatomy. Comparative anatomy is the study of similarities and differences in the structure and anatomy, essentially, of living organisms. So, key terms: homologous structure, structures which are the same or similar, homology, same, right? So, but due to modifications through the course of evolution, may have or serve a different purpose or function than what another organism has the same feature or has for their function. Pentadactyl limb is a five-fingered toed limb in vertebrates, which can be compared to evolutionary relationships. So 5 fingers in humans, and we can see that also in horses. We can see that in the whale, the turtle, the frog, the bird, but very different uses, right? So the bird will fly, the frog will hop, turtle kind of waddles, I guess. Horses run, humans use our limbs also for walking, but also for hands, picking up things, etc. So it supports the ideas that species have evolved from a common ancestor and therefore share similar features, but also that due to divergent evolution, there are particular modifications between each species. Noting that this is a theory. Okay, so this is a theory. It is not based, I guess, on like this is an idea that has come about, and this is an evidence to support that. Comparative embryology is another sort of evidence, or I guess way that they are trying to prove this idea of theory of evolution. So this comparison of the developmental stages of different species, right? So similarities in early development of organisms is strong evidence for divergent evolution. Why? Because it highlights the similar features and structures. So for example, we can see the slits being seen in the early developmental stages of fish, reptiles, birds, and humans. So, however, these develop into very different features. Okay, so we can see here fish, salamander, tortoise. You can see how different they are to the human. We've seen the rabbit, the calf, the pig. Very different, right? But we can see those essential similar features and structures when we first start off with the embryo. We can see here as well: fish, reptile, bird, human, and the slits. Next up, biogeography. So again, we're looking at biogeography. Biogeography, what is this? It's the study of the geographical distribution of living and extinct organisms. So, to support Darwin's theory, he sort of said that new species evolve, right, from a common ancestor when geographically separated from the rest. This is an example of divergent evolution. This can be illustrated through the example of ratites, a group of flightless birds found all over the world who were either isolated when the continents separated, or who flew to their respective continents before developing flightless features. So kiwi in New Zealand, the emu in Australia, the cassowary in PNG, ostrich in Africa, and the rhea in South America. All sort of flightless birds. But they said that in accordance to Darwin's theory, he said, well, that's because of this. He said that essentially what I went through, right? So this idea that they flew to their respective continent and then they developed flightless features, whereas once they didn't have them. So, relative dating. So, this is a form of dating which relies on the assumption that older fossils will be in lower layers of sedimentary rock and younger fossils will be on upper levels or layers as seen in the diagram. I'll show you in a second. So, it does not allow us to determine the exact age of a fossil. Sorry. So, chemical analysis, looking at this, right? So, chemical analysis looks at measuring the amount of a certain chemical, such as uranium, fluorine, in the bones. and nitrogen levels can also be monitored. Stratigraphy, so this idea that this previous concept, which is reliant on sedimentary rock forming in layers which contain the oldest fossils right down the bottom. And biostratigraphy is this idea where fossils, which are known to have existed in a certain period, will be compared to isolated fossils to help determine their age. So there's this sort of trying to figure out what age they're at the moment. So absolute dating, it just enables the age of the specimen to be determined using radioactive elements within the specimen. So it provides us with a quantitative age for a fossil rather than just aging it in relation to other fossils, which we saw over here in specifically biostratigraphy. So fission track dating, so allows us to date a fossil looking at its uranium levels over time. This element can split in a process called fission. Right? So marks called fission tracks can form due to atoms moving at high speeds throughout the substances during this division process. So the number of these fission tracks allow us to identify how much uranium is present and therefore how old the fossil is. So this is used to date fossilized teeth between 5,000 and 100 million years old. Whereas when we compare that to radiocarbon dating, for fossils less than 60,000 years old, the known rate of decay of a radioactive element can be compared to the amount of carbon within the sample fossil. So, for example, the quantity of the isotope carbon-14 can be measured in a fossil to estimate how long the element has been present within the fossil, and then we can go from there and conduct radiocarbon dating. That concludes Module 3 and the sort of summary of Module 3 and the areas consisting around that. Hopefully that was beneficial to you and you sort of understand more clearly what's involved in Module 3 and some of the concepts that are there. We're now going to move into tips and tricks and how to give a Band 6 response every single time. Okay, so let's get right into it. So step 1 is the preparation. So how do we actually prep for this? Well, first of all, you want to color code your syllabus. So red, you don't know it at all, right? So yellow, you kind of know it, but you're still a little bit iffy, still a bit unsure. And green is yes, you know it, you're good to go with it. So then rank your knowledge. So syllabus points, subtopics, and modules. Go through each one, rank your knowledge. How well do I know this? If you kind of know it, how much percent do you know? Like, do you know about 60%, 50%, half of it, whatever it is? And modules as well. Okay, so that is the first step. Second step is to study, right? To make sure you put in the hours. No, you actually really do have to study. So I'll set aside a bit of time at the end to go through a few study tips and tricks to hopefully help you out as well with that. Active recall, so writing down everything that you remember for a specific syllabus point off of the top of your head. So what is involved? How can I improve? So like this is everything that I know for passive transport. And then you can compare it to the notes and to the textbook and go, okay, this is how much I know, this is how much I'm meant to know, how can I change that? How can I actually go ahead and fill the gap of knowledge? So write sample answers for both syllabus points. For the syllabus points. So biology questions are not that far-fetched. And the inquiry questions. So syllabus points, you want to be able to, for example, explain biological diversity in terms of the theory of evolution by natural selection by examining the changes in diversification of life since it first appeared on the Earth. So actually have a response for that one. Second one, analyze how accumulation of microevolutionary changes can drive evolutionary changes over and speciation over time, for example, through the evolution of the horse and the platypus. Next, explain using examples how Darwin and Wallace's theory of evolution by natural selection accounts for convergent and divergent evolution. Have a response for it. All right, so you want to be able to do that for every single one. So explain how punctuated equilibrium is different than the gradual process of natural selection. So how we went through the slides today, right? So for each one we had sort of a response and sort of content that went along with it. This red box, we had content that went along with it. That's what you want to do. That's how you can actually have that active recall. That's how you can improve your answer and go from there. Okay? So hopefully that's making sense. Then you have your inquiry question. What is the relationship between evolution and biodiversity? Then you can answer this entire question using all of this information that you'd gathered from the smaller questions to help you answer the bigger question at hand here. So that is a theory of evolution by natural selection. That's what you're meant to do. And that's a study technique that I would highly recommend. Okay? So making sure that you do understand that. Step 2, how to write a plan. So here's our question. Assess the importance of physical digestion in a mammalian digestive system. So first of all, you want to underline, highlight the key terms like I've done here. So assess is our HSC verb. The importance of physical digestion in a mammalian, specifically in a mammalian digestive system. So don't worry if you highlight the bulk of the question like I've done here, because you are still breaking up the keywords and interacting with what is in front of you. And they're all different. Like they're like different sections that you can then think of differently. Then you want to practice planning questions so that you can find out which planning system works for you. Okay, so which planning system works for you? I've got an example of one down the bottom, which I would highly suggest you have a look into in your own time as well. But yeah, starting off with what is the HSC verb? So if it's assess, we're providing a judgment. And if you are not already aware of the HSC verbs, would highly recommend going onto the NESA website, typing in HSC verbs glossary, and you'll get like a list of what each verb is and the definition of each verb, so you can go about it that way. It's essential. It's very important to understand it. And then go into, okay, what are the key words? In this question, we've got importance as a key word, because it shows, okay, what is the value? We're trying to figure out what the value of it is. How does it actually impact other processes and help the organism? Yeah, so that is where importance comes in. Physical digestion, so mechanical, for example, this idea of tearing, chewing, increasing surface area for chemical digestion and enzymes. So you can see how that's all playing a role here, and we're actually just having little bullet points of information to help us create a better answer. If you were to just go in and just write the answer, you may not be able to structure it as well because you don't have like a plan of a structure going on in the question. And then the mammalian digestive system. So for example, humans. Physical digestion, chemical digestion, mouth, stomach. So you're actually identifying an example and you have the time to think of it in your plan. So a little bit of a recap. Again, I really want you to understand and focus on this. It's very important. Assess. So to provide a judgment, it is essential that you provide a judgment, right? You want to have your own opinion. This is why I always say memorizing is good, but understanding is better. Okay? You want to actually understand the information rather than regurgitate the same memorization that the person next to you has done from the textbook. You want to differentiate yourself from the other people in your class. Importance. So looking at the key term, the second key term, physical digestion, the third key term, mammalian digestive system, and then having points underneath of how important it is and how we can link it to the question and how we can help answer the question. right? So hopefully that's making sense. Righto. So we're going to move on to the next one, which is step 3, which is looking at crafting your response. Okay, so tips and tricks. So here's the example answer for this one. So assess the importance of physical digestion in a mammalian digestive system. So physical digestion is an extremely important process in the digestive system as it increases the surface area of food, allowing processes such as chemical digestion to occur more efficiently. That is our judgment. What is our judgment? It's that it's very important, actually, because we're asked to assess the importance. How important is it? Is it a little bit important? Yeah, half, or is it very important? We've said it's extremely important. It's very important. Why? Because of this. Because it increases the surface area of food, allows processes such as chemical digestion to occur more efficiently. That's why. That's your judgment. So you need to have a decision made of where your judgment sits. That's why I recommend that you actually go in and understand the information rather than just memorize it. Next up, the process of physical digestion begins in the mouth with the chewing and tearing of food by the teeth, enabling larger pieces to be broken down into smaller pieces. This process is continued in the stomach with physical contractions allowing food to be reduced in size further. So we're now going and explaining the process, and we're referring to the key terminology. That's very important. So, key terminology: physical digestion, chewing and tearing, like we talked about, or like we had in our plan. Yeah, larger pieces to be broken down into smaller pieces. This process is continuing in the stomach. Physical contractions, reduced further in size, and then you'd go into SA, surface area and volume, and etc. So you're actually explaining the process. You're not saying, yeah, this process is important. That's it. You're actually going and saying, this is the process. This is why I'm saying it's important, right? And you're referring to key terminology along the way. Okay, next up, further elaboration on judgment. So this is significant in increasing the surface area to volume ratio of the food, allowing enzymes as biological catalysts to break down food at a faster rate. So you're further elaborating on your judgment that you made right up here at the start. So why is it important? Because also of a surface area to volume. But you go into that after you've explained it a little bit, right? So that's where enzymes can come in and they can help break down the food. So you're explaining the process a little bit more. So you're elaborating on your answer. Okay? And then finally, finish off with an example. A nice little example at the end. An example would be the salivary amylase, which begins the breakdown of starch in the mouth. So that's our example of the enzyme that allows food to break down at a faster rate. In that way, we're able to get the 4 marks of assessing the importance of physical digestion in a mammalian digestive system. Okay? I hope that's making sense. Yeah, so I would highly recommend doing this process of breaking everything down into a smaller structure for you to then go into more effectively, I guess. Moving on to step 4, how to think like a marker and why this will secure you a band 6. So it's really important to think like a marker, and the reason that I say this is because when you think like a marker, you know how to write in accordance to what the marker would like to see on the piece of paper. So then you can efficiently and effectively get a band 6 answer. Right, so you want to think, okay, how many marks are actually involved in this? How can I actually achieve this? How do I go about this? All of those questions. So, how to think like a marker and why this will secure you a band 6, right? So try to imagine what the marking guidelines would be for the question that you're answering. So in this case, we're going to go back to our previous question, which was assessing the importance of physical digestion in a mammalian digestive system. So where would our marks be coming from? When we know where our marks are coming from, we know what to write so we can actually get those marks. So we want to be able to provide a judgment statement. That's 1 mark. Judgment statement. What's our judgment? We say that it's actually physical digestion is very important. It's extremely important in the digestive system because it allows chemical digestion, right? And it increases the surface area of food. So we have a judgment, so we can tick that off. Number 2, you want to be able to define physical digestion. Why? Because it's in the question. It's a key term. It's the main term of the question. The question is surrounded by physical digestion and your ability to understand physical digestion. So, the process of physical digestion begins in the mouth, with the chewing and tearing of food by the teeth, right? Enabling larger pieces to be broken down into smaller pieces. Definition. Have we defined it? I think so. We can then tick that off. Because in our criteria, it says define physical digestion. We've done that. Number 3, describe how physical digestion aids with chemical digestion. So we're making that connection with chemical digestion. Let's go back. Chemical digestion. Do we have any chemical digestion? Yes, we do. It's all in our yellow. This is significant in increasing the surface area to volume ratio of the food, allowing enzymes, chemical reaction, chemical digestion, biological catalysts to break down the food at a faster rate. That's 2 marks. How can we actually get the 2 marks? I would say this is a mark, and I would say this is a mark, because you've provided then an example, right? Because we want to be able to understand the difference between the HSC verbs. With describe, you also want to go into a bit more detail because it's worth 2 marks rather than just the 1 that we've been seeing in the judgment and the definition. So an example of this would be salivary amylase, which begins the breakdown of starch in the mouth. That's going to be the 2 marks. That's how you get the 4 marks. So you have to think of that process, and I guess it will become muscle memory when you keep doing it, and it will become a lot more normal as you continue to do the same process again and again, over and over and over. It will just come to you naturally to have all of that. Okay, so let's move on to the next slide, which is looking at how to think like a marker and why this will secure you like a band 6, second part. Secure you a band 6, second part. So things to consider. Does my answer respond to the question and have points for each mark, for each allocated mark, right? So does it actually answer the question? Are you missing a significant chunk of the question that you're not answering? So for example, if you just completely ignore this, then no, you haven't answered the question. You've answered part of the question, so you need to go back and alter your response so that you've answered the full question. Okay? So do you have points allocated for each mark? Do you have points for judgment? Do you have points for definition? Do you have points for that chemical reaction component? Do you have an example? Can you actually go ahead and mark your own response and say, I've given myself full marks because I think I've done enough? If not, go in, alter your answer. This at the start will feel like it's a very long process and you feel like, how can I do this in an exam? But over time, you'll find that this process becomes a lot easier, and you'll find that this process will become like muscle memory. So you won't even need to go through all the steps. You'll just be doing it in your head and go, yeah, that's all good. That's the aim. Next, have I defined all the key terms along with the key terms which may not be explicitly mentioned? So markers are looking for key terms in your answer, and some of the key terms are outside the question. They're not going to be mentioned, but they expect you to have some sort of a reference to those terms. So if we go back to our question, we know our key terms are importance, physical digestion, mammalian digestive system. We've referred to those terms, but we've also referred to some of the key terms that are not listed explicitly. For example, chewing and tearing, which we saw in our plan, physical contractions, stomach, further reduced in size, surface area to volume, biological catalysts, which are enzymes, salivary amylase, so examples essentially, chemical digestion. There's no mention of any of that in the question, but you are expected to refer to some of those terms because that is how you would explain your answer anyway, right? So those things become important. And you may say this is almost impossible to do. You may say that because you're starting out, but honestly, it's not that difficult. I think with practice, it should get a lot easier. Okay? Next one, do I have examples? Okay, it depends on how many marks the question is, but if it is like a 4-marker, you want to include some sort of an example like we have here. Because it actually shows that you've understood the content. That you've gone and thought about, okay, here's even an example for you. Have I avoided a topic, failed to explain a concept in depth? If you have, means you need to go in and alter your response. So, for example, you may have completely removed the chemical digestion component. That means you need to go in and fix your answer. You need to go in and alter your answer so that you have included chemical digestion. So asking yourselves these questions. And again, would highly recommend you start incorporating this process from now. So that when you get to your exams, when you get to the HSC, when you get to trials next year, that this process becomes a lot easier. And it becomes like muscle memory, like I've been saying, like a broken record. So these are the questions you should be asking yourselves before you answer a question, after you've answered a question, and when you are proofreading your answer. So at 3 stages you're asking. When you have your plan, that's before you answer the question. So you're looking at your plan, you can add things there so you don't have to go in and change your response. Even when you're proofreading your answer, you want to make sure, hey, look, have I missed anything? Have I got all the key terms? Highlight the key terms if it helps out. Underline them if it helps out. Highlight the example if it helps. Whatever the case, just make sure that you do have these steps under your belt because they will really change your answer and they will help you out tremendously. Okay, so some study tips that I've put in here to help you all out. So first of all, know what to study. Please look at the syllabus. Mindlessly reading your notes is not studying, unfortunately, and I'm a victim of this as well. Like, don't worry, I think it happens to the best of us, so it's not your fault, but try your best to try and actually study, actually get the work done. Pick up a pen, write stuff down, make it work for you, right? So find out what study techniques work best for you and then go from there. If you're too tired to study properly, watch a good YouTube video that explains a certain concept that you found difficult. Bonus points if you can then go teach someone else that difficult concept. Because when you can teach someone else, it shows that you have your own understanding and you're actually relaying your understanding to somebody else. So it really shows your in-depth knowledge. I mean, it means that you've done well and you're on the right track. Past papers and practice questions are so valuable. So being able to look at those past papers, look at those practice questions from biology, look at what sorts of questions are being asked, identify trends, be able to answer those questions, and then look at the marking guidelines, see where you can improve, continue that process, that spiral again and again will really help you out and will be really valuable for you. So I would highly recommend doing that. Next up, some things that you could do to study. Make notes using the syllabus and other resources, such as the ATAR notes books, the study guides for bio are great. Class worksheets, notes, teacher slides, whatever it is. Have a look at all of those resources, make notes with them, and then relay your understanding. Make your own notes. Get a piece of paper, get a pen, write out your notes of what you've understood. And then if there's certain areas you don't understand, jot them down, highlight them, show your teacher, show your classmates, ask people, ask your tutor, whoever it may be. But try to make sure that you're transparent, you're there, you're wanting to learn and you're putting in that effort. Because those study techniques and study tips that you foster now will continue to grow and it will only help you and you'll only get better at them at the HSC. When you're doing your HSC, when you're doing your trials, when you're going through assessments next year. Teach someone a hard concept. because that shows that you have your own understanding, you're relaying it, like I mentioned earlier. Teaching someone is probably one of the best things that you can do, and it will probably give you a confidence boost. It definitely does for me when I'm able to teach someone else, because it means that I have a good enough understanding that someone else was actually able to learn from my explanation. Past papers and practice questions, like I mentioned earlier, go through that process, go through that cycle of, hey, I've done a practice question, let me look at the marking guidelines. Okay, this is what the marking guidelines say. I'm going to go back to the practice question. I'm going to attempt to do it again in accordance to improving the marking guidelines. Look at the amount of lines as well in the question. If there's 2 lines, don't write 15 lines. That is not ideal. You're wasting your time that you could be spending doing something more efficient and effective for you to get more marks. Palm cards. Have palm cards. It's so important to have palm cards. If you're somebody that uses them, you may not be, but I think it works really well for biology because there's a lot of definitions and a lot of content, so you can have like the definition and then the term and the like concept at the back, something like that, if that works for you. Draw pretty diagrams. I drew diagrams. They weren't necessarily very pretty, but they were there and they were really helpful for me to break the sort of notes that you have that is just like really boring and just that one repetitive note taking that can, be quite boring for your brain and cause you to switch off. If you have something different that your brain can use to understand and absorb information, that can be really helpful. Record yourself explaining things and then listen back on it later. So whether that be on a video or audio, just so you can then go back and keep listening to it and actually develop more of an understanding, because repetition is key to success in a lot of these courses, especially when you're memorizing a bunch of stuff. This can also help you understand it rather than just stop at memorize. Okay, so those are some things. Again, making notes, writing out important concepts, teaching someone, really important. Past papers, HSC practice questions, looking at the marking guidelines, going through that spiral of improvement. Palm cards if that's something you're interested in. Pretty diagrams, again, if something you're interested in. Recording yourself as well. So those are some of the tips there. Another tip is to find your optimal study time. Everyone has a different schedule. Everyone has different times where they feel most productive. Some people may feel really productive in the morning. Some people will feel really productive during the nighttime. It is your job to find out when you feel most productive and you feel like you can get the best work done, to find that time and use it, right? Rather than waste it. So you have to identify your available hours to study and set them aside. You may have extracurricular activities such as sport, whatever it may be. That's completely fine. Just make sure that you have time set aside for study as well. Set aside times where you have an opportunity to be productive and you won't be interrupted by anybody, because when you're interrupted, whether that be by your phone, whether that be by a family member or a friend, whatever it is, you will break that that cycle of studying, and then you probably won't come back to it till later, and you may not have the same mindset, the same focus that you once did. So try to find a time where if you're doing something, you can do it continuously for a while, so that you can try and be as productive as you can. In the lead up to your study hours, hype yourself up. Something I did was actually reward myself at the end. So I'd say, okay, I'm going to study this week, and then on Saturday or Sunday, I'll go out with my friends or with my family for lunch or for dinner or for a coffee or whatever it is. So in that way, I'm actually hyping myself up. I'm actually like, you know what? Yes, I'm going to push as hard as I can. At the end of the week, I'll get a chance to enjoy. Okay? Study timetable. So I'm sure every study skills talk that your school has made you listen to has said this, but my study timetable was how I survived the HSC. So make it early. Plan for every single thing, right? You don't need to do this before the exam. You need to do this before the exam, and it needs to be done in time so you don't stress about missing anything. So even the tiniest thing, like I need to actually analyze, how to approach this biology question. Okay, let me note that down. Hey, I actually need to do this for English, right? Study timetable is for all your subjects. You can not just delete something off of the study timetable if you don't get something done on the day, then change it to another day so you still have time to do it, and do it immediately after it's been done. So if you haven't been able to get through writing a practice question for the biology practice question that you were doing, because you got delayed at school and you actually have an extracurricular on at school, change it. Okay, I'm actually going to move this to Friday evening. Right, so you're not just deleting it or completely removing it. As soon as you come home after that extracurricular, you actually have time and you go, I'm going to do this on Friday. Or I'm actually going to spend a bit of time now that I was actually going to have free time for because of that time that I've cut off. I know it's annoying and I know it's a little bit like, can't be bothered, but please find the time. You will thank yourself when you get to that exam season and that exam stress comes to you. Next up, know what to study. So look at the syllabus. Yeah, we talked about that. That's fine. Okay, reading time. So read through the entire paper briefly when you have an exam. Choose which question you'll do if you're given options. Think about some of the approaches to any trick questions that you see. And don't overthink any questions that you think look hard. Chances are they're probably there to trick you, to make your brain go, oh my god, I can't do it. But when you actually read it, try and focus, sit down with the question, look at the diagrams, whatever's given to you, and just really analyze it, and then you can have a go at answering it. Okay? That is what your reading time is for. To actually have a moment with it before you just go in straight away into writing. So, a lot of the time in multiple choice for a lot of subjects, they actually provide a recommended time that is way higher than what is, I guess, normal or realistic. So that was especially the case for me. So I usually left like the extended response things towards the end. And then if I sped through, my previous sections that I could have leftover time to use to perfect my essay. So always try to leave at least 5 minutes at the end of an exam to double check over your work, and that check if you haven't made any major screw ups. And how do we do that? Let's go back. Answer these. Answer them, make sure you know them, make sure that you have enough practice that you have memorized them when it comes to exam time. So always try to at least have 5 minutes at the end of an exam to double check over your work and to check that you haven't made any major screw ups. If you can't do a question, highlight it, and in your extra time, prioritize those highlighted questions before going through everything else. And that concludes today's lecture. So I hope that you found this useful. I hope that it was helpful. And yeah, I hope that you learned something today. Other than that, I would like to say thank you so much for taking out the time of your day to listen to this lecture, and I hope that you gain some valuable information and some study tips that will help you and that you will utilize. Other than that, thank you so much, and I will hopefully see you all soon.